the other day we started with the chapter cell. The first chapter we started. Cell. The fundamental unit of life. And last class, I think we already finished discussing about unicellular, multicellular organisms. So prokaryotic, eukaryotic, did we discuss? No. So last class, we must have already started the concept of cells. What are cells? And we must have already said that cells are the functional unit. Cells are grouping together to form tissues. And that is how an organism gets formed. That is how the complexity of an organism, etc. And if we have already studied about the concept of unicellular, multicellular organisms, then what next? We understand that unicellular organisms, only one single cell, but that cell also undergoes all the variety of functions, all the variety of physiological functions. That cell also undergoes all the variety of physiological functions. And when we are talking about if it is amoeba, always to understand that amoeba also needs to undergo the process of nutrition, excretion, etc. So, did we understand that in case of a single-celled organism, can there be anything known as division of labor? No, there cannot be anything known as division of labor. And if it is a multicellular organism like us, or the plants, or any other insect, or any other amphibian, then there is something known as the division of labor. What was the meaning of division of labor we had understood?
Different cells perform different functions in specialized tissues.
Different tissues perform different functions, different organs perform different functions. But this kind of features we never come across in a unicellular organism, of course, because the functions are so simple, all carried out by the cell itself. So this is what we had studied the other day with examples. Now, the next concept that we are going to talk about today would be prokaryotic and eukaryotic cell. The other day we had studied how can organisms be complex or simple. Now we try to understand how can cells be complex and simple. Prokaryotic, if I talk about a prokaryotic organisms, I would say that these prokaryotic organisms have a single cell, and this coil structure that I am drawing, this coil structure is what is known as the hereditary material here, and this hereditary material we would name it as the chromatin fibers. This hereditary material we would name it as the chromatin fibers. So what type of an organism I am drawing here? I am drawing the cell of a bacteria. I am drawing cell of a bacteria here. And parallelly, I also would draw an animal cell. Say suppose I draw an animal cell here. Now when I am drawing an animal cell, then how should I draw the nuclear region? How should I draw the chromatin fibers, the chromosomes? To draw the same chromatin on the chromosomes, I first of all have to find out that how should I arrange those chromosomes or the chromatin? Can I arrange them same like this? No, they cannot be arranged same like this, because now I am talking about these are the chromatin fibers, but additional, this group of organisms also have something known as the nuclear envelope or the nuclear membrane. And if that is so, then such type of organisms or such type of cells, not organisms, such type of cells are known as eukaryotic. So what did I tell you just now? If I come across a cell which has, the chromatin fibers, but the chromatin fibers are just scattered in the cytoplasm, they are not wrapped up by any membrane, they are not protected by any membrane, they are not covered by any membrane. Did I see any membrane here? Do we get to see any membrane here? No. And on top of that, the chromatin fibers can be just one or two, not more than that. The chromatin fiber would be just one or two, and the chromatin fiber, those one or two fibers would be extremely long and extremely coiled. One single thread, extremely long and coiled. Yes, this particular region is known as the cytoplasm. This particular region is cytoplasm, but this cytoplasm, we will hardly get to see any parts present, which we call them as organelles here in the animal cell. So if I talk about the cytoplasm, here is the cytoplasm vacant like this, clear like this? No. Here there will be a large number of some organelles known as lysosomes. Then we will come across the mitochondria present. Then we will come across the endoplasmic reticulum present. Okay, so all these organelles are here. Then we talk about the centrioles present, and then the ribosomes present, and many more. But if I talk about the cytoplasmic region here, the cytoplasmic region is almost vacant, clear, with just few vacuoles and just few ribosomes to produce proteins. Only that much. But the most important thing that we have to take a note is the nuclear membrane, absolutely absent here. So here there is nothing known as nucleus and cytoplasm. Rather, these chromatin fibers which are scattered in the cytoplasm, we will name them as the nuclear region. We should name them as a nucleoid. We will name them as the nucleoid. What about this side? They have got the nuclear membrane, they have got the chromatin and the chromosomes also well developed. So we have to name them obviously as nucleus, and we cannot term them as nucleoid anymore. So now that we are talking about the characteristics of a prokaryotic and the eukaryotic cell, then what are you supposed to understand as major differences? What are you supposed to understand as major one or two differences to write? Number one, if it is a prokaryotic cell, by the term pro means primitive. Absolutely simple. Now these primitive cells, how do we say that they are primitive? By looking into the nature of the nuclear region. Look into the nature of the nuclear region. The nature of the nuclear region makes us understand whether the cell is simple or primitive. If the cell has got nuclear membrane and if the cell has got all the organelles, then it is an advanced cell, something like this. Eukaryotic, U means advanced. U means advanced here. And when I talk about pro means primitive here. Okay. Karyotic, when we talk about the word karyotic is derived from the word karyon. Karyon means nucleus. How complex the nucleus is, how simple the nucleus is. Okay. So these are the two main differences that you will always remember. Apart from that, there are other differences. I think the characteristics of the shape, if I have to understand. Yes, the shape. If it is a prokaryotic cell, obviously they are smaller. If it is a eukaryotic cells, they can have a variety of shapes. As it is, they are bigger, larger. What else? The functional property, obviously, if it is a eukaryotic cell, they will be more complex in their functions, a type of enzymes, proteins, etc. If it is a prokaryotic cell, obviously, they will be releasing just one or two types of proteins, and the functions also will be very simple. Look at the volume there. Look into the volume there. There are some differences between the prokaryotes and the eukaryotes. I want to just read them. Just read those differences. Just read those. Go ahead. Go ahead. Prokaryotic and eukaryotic. So, diagrams you should be able to identify for prokaryotic and eukaryotic. You may ask to draw diagrams also to distinguish. So you are not supposed to ask so elaborate diagrams. Elaborate diagrams you are not actually supposed to draw. You are not asked to draw. But the basic difference you should be able to show that if it is a eukaryotic, well-developed nucleus it should have. Prokaryotic will not have. Only this feature for you all is enough. Okay. So what are some of the main differences? See there, tick those points that you should always study. The first difference I say that if it is a prokaryotic cell, it will always have only one single chromatin. One single chromatin, or at the most two. It will be one single chromatin which is extensively long, thin, coil. But if it is eukaryotic, it can have a variety of number of chromosomes. It can have hundreds of chromosomes, thousands of chromosomes also. Now if I talk about even ourselves, say, even if I talk about ourselves, we have 46 chromosomes in our cells. So there are so many other organisms which have many more chromosomes than us, and some organisms of course lesser than us. No? So if I talk about the developed group of organisms like us, the number of chromosomes are variable. Variable number of chromosomes there are. What else you are supposed to understand? If it is a prokaryotic organism, the nucleus is unorganized. And what is the name of that unorganized nucleus? Nucleoid. Underline if it is there mentioned. Nucleoid. Read those points, you will find. It is nucleoid. Unorganized nucleus is known as nucleoid, and when I talk about the eukaryotic cell, they always have a developed nucleus. We talk about the cell organelles now. Another point of difference. If I talk about the prokaryotic cells, cell organelles are almost absent. Cytoplasm is almost clear. Cell organelles are almost absent, except for they have got some few ribosomes. Except for they have some few ribosomes. Now the ribosomes are meant only for synthesis of some few proteins. And if I talk about the eukaryotic, for eukaryotic, we understand just now as I have drawn this block diagram also. For eukaryotic, there are a whole lot of organelles for different type of functions. Okay, so take those 2-3 differences, and you should be knowing those differences whenever you are asked to write. Those differences you should know. Now, this is about the prokaryotic eukaryotic cell, unicellular, multicellular organism. So that's the first part of the chapter. Now we start talking about the animal and the plant cell and their differences and their features, characteristics, etc. You take out your pencils, take out your notebook. First of all, we'll draw the diagram of the animal cell and then learn the organ one after the other. Whose scroll is this? Draw it fast, huh? It's a simple diagram. We have to practice 2-3 times, then you will always get it.
Why too small? Why is it so small? How will you label the parts there? Your diagram should be not too dark but prominent. How you should draw the diagram? Now see, for this diagram drawing that you have in your syllabus, 2 to 3 marks in this diagram. Huh? Now if you want to get the complete 3 marks there, then certain things of course you'll have to remember. The diagram that you will draw, you will not draw with a very light pencil. At the same time, the outer membranes of the diagram, whichever diagram it is, because tissues and all also that chapter you draw many diagrams. So when you draw the diagrams, make it a point that the membranes that you draw should be dark, prominent. Okay. Then when you draw the diagram, should not be too small or too big. It should not be page full also. Half a page is maximum, not more than half a page that you should draw. And you will always try to draw the diagram on one side, meaning towards the margin. So just 1 minute. See, I get to see that she also has done this way. You are supposed to draw the diagram a little bit towards the margin and keep more space this side on the right side. Very good. On the right side. So that you get space to write down the labels and try to put most of your labels on the right side. Labels all with pencil, drawing all with pencil. No color, sketch pen, nothing. Okay? So draw big.
So must we draw then?
Huh?
Should I draw it?
You should, I think so. But don't take so much time. I cannot give you so much time. Take it fast. You loved art in your lower classes?
No, I drew a lot in my childhood.
Huh?
I used to draw a lot in my childhood.
Then why is it so difficult for you to draw?
But since then I haven't drawn it.
Hmm?
I haven't drawn a lot since.
Draw it big. See, again you have taken it small. Half of a page, I told you. Half a page. You have taken it small already. Half a page. Not more than half a page. That should be the limit. No. Again, you are taking. This is not a diagram. You will not get more than one for this diagram. Your effort is gone. 10 minutes you will spend, but not more than one. Or with pencil. Half a page you take now. At least 8-9 lines. Take that margin. 8-9 lines. How much it should be? Count 8-9 lines. Okay, so that much it should be. So you get an idea how big my diagram should be.
So now we are talking about this animal cell. Okay, I think most of you have finished drawing. So when we are talking about, this is the diagram of animal cell that we have drawn. Now we are going to learn the parts and their characteristics, their functions. So let us take one by one. We are talking about the animal cell and we are going to learn their functions. So as I talk about the organelles, I'll talk about the organelles one after the other. What are organelles? What are cell organelles? Always remember the parts of a cell which perform various functions. Just like our body, if I talk about our body is made up of so many organs, and every particular organ has a specific function. No? Same like that. The organelles that we talk about inside a cell are the parts of the cell. I last time also must have told you that the moment we talk about multicellular organisms, we have got the heart for a particular function, pancreas for a particular function, the kidney for a particular function. Same that way, you will find now that the Golgi has a separate function, the mitochondria separate, the nucleus separate, the endoplasmic reticulus is a separate function. So these are the different organelles. That's why they are named as such. We first of all start with the cell membrane. Let us start with the cell membrane. Now, what is cell membrane? It is a limiting wall that we understand. The cell membrane is a limiting wall. What are some of the structural features? What are some of the structural features, if it all some special property this particular structure has? When we are talking about the cell membrane, it is composed of, made of lipoproteins. It is made of lipoproteins. What is the meaning of lipoproteins? Lipo from the word lipids. Lipids means fats, fat molecules. So that's the reason, again, we should be always containing an adequate amount of good fat in our diet. No? That's the reason we should always contain adequate amount of, we should always have adequate amount of fats in our diet, so that even if there is a torn out functions happening, damaging functions happening in our body, the cells are repaired very fast. Repairing of the cell always means formation of the membranes, and for the formation of the membranes, adequate amount of good fat is required. So now when I talk about the lipids, lipids are a type of fat that we get from our diet, from our food. And proteins, you already know, variety of different type of proteins are cell producers also, and we need to get them by diet also, so food also. But now I'm trying to understand that this plasma membrane that we are talking about as a single thin wall. Now it looks like a single thin wall, but actually it is not. The single thin wall, when I talk about, look into the detailed structure. The detailed structure will something be like this. The detailed structure will something be like this, where there are two layers of fats. And out of these two, or in between the layers of fats, what are they? Now these are the two layers of fats that I'm talking about. And in between these layers of fats, there are some proteins arranged. Some proteins will be like this. Some proteins will be spanning the entire width like this. Some proteins would be spanning the entire width like this. And some proteins would be just absolutely on the bottom layers, and we cannot see them from the top or from the outer surface. So this arrangement, I will tell you, is comparable to certain type of flooring actually. Now I'm talking about this green structures that I've drawn as proteins. And what about these red two layers? These red two layers are the lipids, the fats I told you. And this particular structure is known as the fluid mosaic model, or the fluid mosaic structure of the plasma membrane. Now what is the meaning of the word fluid mosaic? Now when I say fluid, you understand it has a semi-fluid consistency. That is what we understand. Fluid. It is not a solid structure. It is not a tough solid structure at all. Fluid, soft. What about the word mosaic? Mosaic means, I think sometime or the other, all of you must have seen, or you may be having in your homes also, the mosaic floors. Now, mosaic floors, are you aware what are mosaic floors? Especially nowadays, we get to see much lesser. But if I talk about some buildings which are some few years back constructed, and even some hotels and etc., we get to see some flooring which is made of a white cement, and then some marble chips. You can see the different colored marble chips, some red and some brown and some yellow. Those marble chips you can see.
So it is understood that those floorings, how it is done, they would put one layer of cement, then one layer of white cement is there, then they would sprinkle and throw those marble chips, and then one more layer of white cement, and then they would polish. So some proteins or some marble chips are completely embedded, which you cannot see from the top. Because some has all completely gone down. Some partially only you can see because they are mostly embedded and only little bit of it is there. And some quite a lot perhaps you can see, some quite a lot perhaps you can see, some quite a lot perhaps you can see because they are small and no. So they biologists believe that when we talk about those mosaic floors and the making up of the plasma membrane is quite similar. There's a variety of proteins but differently arranged. There are a variety of proteins differently arranged and two layers of lipids, almost like two breads in a sandwich that we talk about. No? So that's the reason it is also known as the sandwich model also. Fluid mosaic model, that is the basic characteristics always to be talked about when we talk about the plasma membrane. Okay. And the composition already we have understood. What is another special property that we have to understand about the plasma membrane? The plasma membrane is semi-permeable. Now this is a very, very important property that you have to remember. Plasma membrane is semi-permeable. What is the meaning of this? Semi meaning? You understand? Partially. Permeable meaning?
Allows objects to move
Transport. Allowing transport. So the plasma membrane may allow transport of certain molecules in and out. And again may not allow the transport of certain molecules in and out. So there may be some restrictions. No, there may be certain type of molecules. Now I talk about certain type of molecules are there, and here certain type of molecules are there. But maybe these type of molecules are going out and these type of molecules are not going out. If this is what we are talking about, then this is semi-permeability. Same for when you talk about entering inside. Same for when you talk about entering inside. Certain molecules only can enter inside, some certain molecules cannot enter inside. Now, this is only because of certain arrangement of proteins and lipids actually, and the functioning of a cell also. You are getting questions often in the paper. Explain the semi-permeability feature of the plasma membrane. Then what are you supposed to write? Semi-permeable nature of the plasma membrane means it allows certain molecules to enter or exit, and certain molecules not to enter or allowed to move out. So this is a simple concept sometimes for 1 mark, 2 marks you get. Semi-permeability of the plasma membrane. What are some of the functions? If you have to look into certain functions of the plasma membrane, now let us look into some functions. What do you think can be one of the most important function of the plasma membrane? It holds the cell contents. Yes or no? It holds the cell contents. When I say it holds the cell contents, meaning this particular membrane, though it is not very tough and rigid, but it is just like a wall. And if 2-3 cells are nearby, the contents are not flowing out and mixing with one another. Now, the moment there is a small break also in the membrane, the moment there is a small damage, there is a small part of the membrane torn also, do you think this cell will be able to function? Will this cell be still considered as a functional cell? No, because so much of the enzymes and proteins and cytoplasm will start flowing out. The moment there is a tear, a torn or a wear or a tear happening at any part of the plasma membrane, a small one also, will allow the oozing out of the cytoplasm, coming out of the cytoplasm and the enzymes and the proteins, etc., and this cell will not be able to perform the function that it is assigned to. So that's why we say that yes, it is holding the cell contents, and at the same time, it is also providing protection. Now, what type of protection the plasma membrane can be giving to a cell? What type of protection it must be rendering to a particular cell? Protection, yes, because quite a number of types of substances and molecules are not allowed to enter. Unwanted germs, pathogens, excess amount of water, not allowed to enter. So if that would have been allowed, then maybe the cells would have damaged in a short period of time. No, maybe. So when we are talking about protection, yes, the cell membrane does not allow so many different types of substances to enter. Excess amount of water does not enter. So that way the plasma membrane allows a proper functioning of the cell also. And third is, it also provides a definite shape to the cell.
It also provides a definite shape to the cell. With reference to semi-permeability, what function are we supposed to write? Brings about transport. Certain type of molecules which are produced in one cell can go to another cells, and molecules made in other cells can enter into a cell. So a particular cell can allow movement in and out. It can go, it can come in. It can go, it can come in. So transport of molecules in and out of the cell. Transport of molecules in and out of the cell is also a function of plasma membrane. Transport of molecules in and out of the cell. So this is about the plasma membrane. Now, how are you supposed to study the organelles? Whenever you study the organelles, I've just finished with one. How are you supposed to study the organelles? Don't look into the entire chapter, or don't look into this concept as a big entire chapter. There are just 4 things to study in this chapter. Number 1, unicellular, prokaryotic, eukaryotic. So that was one concept that I studied, that I taught you here. Unicellular, multicellular, prokaryotic, eukaryotic. Only this one. Now we are talking about what? Cell organelles, plant and animal. This is what we will study. And apart from that, there is another concept known as membrane transport. Something related to cell membrane permeability that I introduced just now. Something related with that. You don't want. So only these are the few contents of the chapter. So how will you read the cell organelles? What is its structure? And I will tell you, don't go into any other elaborate textbooks or reference books. That is not necessary at all for you in the 9th standard. Even in the 10th also you don't need. So mostly you are, because when it is a time for exam and doubt solving, students will come with those reference books. You don't need it all. You don't need it all. You will study specific what is in the syllabus. Of course, for knowing and understanding, you can read anything. There's no limitation at all. But for the exam, you are basically to understand the structure, few basic concepts about the structure, and 2-3 functions only, and detailed diagrams we have to learn how to draw. Okay, so this is how you will study. Not to panic, there's nothing elaborate that you have to study here. So this is about the cell membrane that you will study. Is there any diagram drawing? Nothing. About the cell membrane, there is no diagram drawing at all. Only these concepts you have to study. Basic 2-3 points about the structure that I told you just now, and 2 or 3 functions. Next is we move on to the next important organelle, that is mitochondria. What is mitochondria? Did you ever read about mitochondria earlier?
Yes.
What does it do?
It is the powerhouse of the
Why is it known as such?
because it performs the, what do you call it?
What is
It's basically, it gives the cell food for the cell is basically done in the mitochondria. It's similar to that function that leaves perform for the plants. It basically gives the plant the cell protein.
No,
It provides energy. Yeah,
energy.
Yeah.
So that's why I told you, when you read, you will read basic specific things. Okay, so 9th and 10th, very simple, provided you know what to study, how to study, from where to study. And you sit with 10 books and you yourself don't know what to study. No, so that should not happen. So now, when we talk about mitochondria, very simple to understand. What is mitochondria? It is a cell organelle. What is it supposed to do in the cell? Provide energy molecules. Provide energy molecules. Produce energy molecules. But what is the form of the energy molecules we will learn? Energy molecules, something like ATP. ATP. You have already come across ATP? No? Okay. So let us first of all learn the structure of mitochondria. What is the structure of mitochondria? Now when we draw the mitochondria in detail can be drawn this way, where the outer membrane just now I drew very, very even. And now that I draw the inner membrane is something like this. It is folded like this. So now I'm drawing that this is inner membrane. And the outer membrane is nothing exceptional about it. I'm talking about the outer membrane, which is very, very simple and very, very even. This is the outer membrane. So if an organelle is having two membranes, then we will say this cell organelle has a double wall. It is double walled. Cell organelle is double walled. What do you understand by double wall? It must be having two walls. Two distinct walls it must be having. So now that I talk about mitochondria, yes, it is double wall because it has got two walls. What about this? When I talk about the inner wall is folded. So when I talk about the folds of the inner wall are known as cristae, plural. Or if I have to write only one, I should write crista. And if I am naming one or two together, then I should be writing cristae. So what did I tell you just now? That this mitochondria that I am talking about is a double-walled cell organelle. It has got two membranes. The outer membrane is even, smooth, even. And the inner membrane but has got many finger-like projections, many folds it has. And each of those folds, if you have to name, we should name them as crista, and a number of them together, cristae. Now, what is so special about this crista or cristae that we are talking about? On those crista are present some knob-like structures. On those crista are present some knob-like structures, and these knob-like structures are known as the F0 particle or F0 elements. I'm giving you both the words, just check what are written there. F0 particles or elements. F0 elements also they are known as, most commonly known as F0 particles. So what is happening in F0 particles? These are the sites where the process of ATP synthesis happen. This is the site where the process of ATP synthesis happen. Now the question is, what is ATP? When I talk about ATP, ATP stands for adenosine triphosphate. ATP stands for adenosine triphosphate. What is it? It is the chemical substance that the mitochondria produces, and this chemical substance, when it is broken down, provides a lot of energy to the plant cells. That is the reason the mitochondria is known as powerhouse of cells. Now, why should the mitochondria gather so much of energy? Why should the mitochondria provide so much of energy? Why should the plant cells get so much of energy? It doesn't have to talk, it doesn't have to run, it doesn't have to swim, it doesn't have to laugh. Why so much of energy? But can you tell me, the plant cell, should it require energy or not? Does it require energy? Should it require so much of energy, or should it? It must be requiring energy, or it should not be requiring energy? It should, yes. For functions?
For the plant survival, for like the functioning of the transport of food
and transport.
water.
Now, yes, of course, the energy requirement is much lesser than us, undoubtedly, but yes, the plants also require energy. You know, the mitochondria, a whole lot of mitochondria in every cell, producing so much of energy. But why? Because one major function that the plant requires energy for is the transport. Talk about these tall plants. Those betel nut and coconut and palm trees and etc. So many tall plants are there with a height of, there are many more also if I talk about the pine and the oak and all this. There are so many plants which have got a height of something around 30-40 feet. And can you imagine the transport of water right from the soil to those heights without any pumps, without any motor? Can you imagine the water driven from our tanks to those 8-9 floors? That only requires so much of energy. It's not so. And can you imagine water driven to those 30-40 feet high without any motor, any machines? So that efficient, that means they must be requiring so much of energy majorly for the process of transport. And also now, transport of food also. No? Transport of water, just now I told you, transport of water from the soil, and also the transport of food from the leaves to all parts of the plant. So that is a major function in the plants why they require energy. And now if I'm talking about the animal cells, there are so many things to mention. Why should we require energy? Can you give me 5 reasons why our cells should require energy?
Again, for the transport of
okay. Transport of the blood and the nutrients, okay. And Why should we?
Walking,
Ah,
like basic life functions.
contraction of the muscles, movement. We constantly show movement. It's not so. Now remember, the moment we are constant, the moment we talk about movement, even now when we are sleeping, we are changing our size. That time also so much of energy we require. No, so how do we understand? Because whenever we are sick and down with fever and a massive infection, we're not getting that much of energy to turn sides also. No, somebody has to help us no, at that time. So now that we understand that when our body is normally functioning, and if our body is providing so much of energy, then we have got energy for everything to do. No, so mitochondria present both in the plants and the animal cells, providing so much of energy. Now, what is the energy? You will get questions in your paper. Name the energy currency. Currency means what do you know? What is the meaning of currency?
Something we use in exchange of goods.
Aha, she talks about gold, she's straight away going to gold. Now, when we are talking about currency means some unit, I can say, unit of money. Okay, so unit of money, if I say, yes, now in case of mitochondria, if I say that, what is the unit of energy that they are producing? What type of energy molecule? Energy in the form of ATP. Please write it down. ATP. Now ATP, just the full form you write. Points you will write when I write it down. ATP. So when we are talking about ATP, full form adenosine triphosphate, and that is the form in which the mitochondria always produces energy. That is the form in which mitochondria always produces energy, and this is a form of energy which is constantly required by our cells, plant cells, to bring about various different type of functions. So first you just write down ATP stands for what? ATP stands for adenosine triphosphate. ATP stands for adenosine triphosphate. Now let us learn the structural features also again. I said mitochondria is double walled. This is one feature. What else? Mitochondria is double walled. Apart from that, the folds of the inner wall. Folds of inner wall. Folds of inner wall. What are the folds of the inner wall known as? I told you. If I take the plural term of itself, the cristae. The folds of the inner wall known as cristae. And next is the knob-like structures. This is what we call it as the knob-like structures. This is something like the studs. So we talk about the knob-like structures. Knob-like structures on the cristae. Knob-like structures on cristae are the sites for ATP synthesis. Synthesis, you know, no? What is the meaning of synthesis? Formation. Huh?
Yes.
Synthesis means formation. So are the sites for ATP synthesis. So that is about their structure. One more point still to understand when I am talking about the structure. If I talk about the other cell organelles, we will never generally understand that apart from the nucleus, there are just two organelles which have their own chromosomes and own DNA. Nucleus, we basically talk about nucleus and associate nucleus with chromatin and DNA, is not so? But if I have to talk about mitochondria, now the diagram will be complete. We should also draw something like this. We should also draw something like this, where I am saying, what is this? This is the DNA of the mitochondria, also termed like this. What is the meaning of writing it this way? Mitochondrial DNA. Mitochondrial DNA. Nucleus is supposed to have DNA. Is there any other organelle which is supposed to have DNA? Yes, mitochondria, both in plant and animal cells. And if I talk about chloroplast, again in plant cells only. These are the two organelles which have DNA of their own.
What is the function of the DNA? Like, why does it differ from the nucleus?
Because now these are very smart organelles. They don't want to depend completely on the nucleus. They want to carry out certain functions on their own also. So now these two specialized organelles, they have their organelles, like they have certain structures known as the DNA of their own. And since they have DNA of their own, they can produce certain proteins of their own and they need not depend on the nucleus. And that's the reason in a newly formed cell,
they can very fast and very rapidly undergo the process of duplication, formation. Whereas for the formation of the proteins and functions of the other organelles, they're absolutely dependent on the nucleus. The nucleus will give them commands. And then the endoplasmic reticulum and the lysosome and the Golgi, etc., they will function as per the indications and instructions by the nucleus. The mitochondria does not want to function absolutely under the instructions of the nucleus. It will partially. But partially it will keep on producing its own functions, producing its own proteins and enzymes and performing its own functions and bringing about its own multiplication from one mitochondria, new mitochondria can grow. So that is what is this particular specialized feature of mitochondria. It has its own mitochondrial DNA. Function, what is the function? It is known as the powerhouse of cells. It is known as the powerhouse of cells. You will be asked questions in the paper. Give reason, explain why is mitochondria known as the powerhouse of cell. You will have to write a proper answer. It is known as the powerhouse of cell because it produces energy in the form of ATP. The energy is necessary for breaking down food. No, breaking down food and providing energy again. So breaking down food means what? Let us talk about mitochondria in our own cells. What is the job of switch on? Now, when we are talking about mitochondria that we have in our cells, what will be the job of mitochondria in our cells? The mitochondria in our cells will constantly release energy, ATP. That ATP will bring about the breakdown of food in other cells, all the other cells. So whatever food breaking we are talking about, the glucose which are present in every cells, the glucose molecules present in large numbers in every cells. Are you at all aware that when I talk about how do we get energy? When we eat a lot of carbohydrate food, we get instant energy. A particular day, you are feeding only on fruits. You absolutely don't take any fats or carbohydrates. You absolutely don't take any fats or carbohydrates. Your energy level comparatively will be lesser. You may not be suffering from diseases. You may not be suffering from any other ailments, but your energy level will be comparatively low. What does that mean? Whenever we are talking about food rich in glucose and carbohydrates that we consume, this glucose and carbohydrate enters into our body cells by the process of digestion. And now that this ATP released by the mitochondria, this ATP also enters in our cells, the glucose also enters in our cells, and this glucose gets broken down and energy molecules are released. Energy molecules are released in our cells with the help of this ATP. So that energy again helps us to, that energy is also in the form of heat and ATP. So that energy helps. What are we writing here? We are writing that why is mitochondria known as powerhouse of cell, or what is the role of this ATP? ATP will always help in the breakdown of glucose. This will always help in the breakdown of glucose and provide additional amount of energy. So this is the explanation for this. We will write answers later. Next is, what is the other job of the mitochondria? Helps in the synthesis of variety of enzymes and proteins, because it has got its own DNA. Enzymes and proteins to bring about or carry out oxidation of food. This is what I was explaining you just now. This is what is known as oxidation of food. What is meant by oxidation of food? Breakdown of food to release energy. Again, just one more concept to tell you, that if I talk about breakdown of food to release energy, again I said yes, that's the reason carbohydrate food is necessary. But if I understand that we are consuming so much of carbohydrate rich food, but our body does not have adequate amount of ATP. We are eating carbohydrate food, but body does not have adequate ATP. Then also, will we get adequate amount of energy to do the different type of activities? No, that's again difficult. That's again difficult. Why? Glucose has to be broken down. So what am I talking about? I'm talking about oxidation of food. So what is the meaning of oxidation of food that I told just now? Oxidation means, yes, this in the presence of oxygen
in the presence of ATP should be broken down to carbon dioxide and water and a whole lot of energy again. So we are dependent on that energy. We are using this energy produced by mitochondria. Okay, this we will learn again later. So what are the roles, what are the functions of mitochondria we understood? Mitochondria, now check those functions of mitochondria that are there in the volume written. Look into those points there. Look into those points there, mitochondria. Could you find the functions of mitochondria? Read and see if it is understood what we just now discussed, if there's anything that you don't understand.
It's just the
Same thing. Any word that is not understood, is it clear? Functions. So two functions you should be understanding. Two functions you should remember. So this was mitochondria. Now next is we'll start with another cell organelle, that is endoplasmic reticulum. Now look into this tube-like structure. Can you see this tube-like structure that we have drawn here? One feature of this tube-like structure that you draw also, we should always start from the nuclear membrane. They should always start from the nuclear membrane, unorganized tubes. And are they double walled? Look into a single tube. Are they double walled? Yes. When you talk about these tubular structures, no, they are 1, 2, 1, 2, 1, 2. Look at this part. Are they double walled? Are they double walled? This part you look at it. Are they double walled? Yes. Now, what is this particular organelle about? It is a network of tubules starting from nuclear envelope and quite a number of times touching the cell membrane, quite a number of times may not touch the cell membrane. This is what you will remember when you draw also. Sometimes these tubular structures have got some other organelles attached to them. This is another organelle altogether, ribosome. Sometimes endoplasmic reticulum has ribosomes attached to them, sometimes not. So if ribosomes are attached, then we would say that this is rough endoplasmic reticulum. And if ribosomes are not attached, we would say this is smooth endoplasmic reticulum. So this is a question always you are getting in the paper. Name the two types of endoplasmic reticulum. Now what to write? Rough endoplasmic reticulum, smooth endoplasmic reticulum. Now, when I talk about the rough endoplasmic reticulum, in what type of cells it will be present? The rough endoplasmic reticulum has a specific function, and that is why they are majorly present in muscle cells. They are present majorly in the muscle cells, and especially certain few cells of the body which show rigorous movement, extensive functions which show. And if I talk about the other side, if I talk about the smooth endoplasmic reticulum, the smooth endoplasmic, does it have this organelle attached? No, they don't have. Since they don't have, their function is also separate, and that's the reason they are also found in specific cells like the liver cells. They may not be found in the muscle cells in large numbers, but they are specifically found in the liver cells. They are found in the cells of the stomach, cells of the intestine, etc. Now, why like that? Here, we are talking about two types of endoplasmic reticulum, the rough and the smooth. If I talk about the rough endoplasmic reticulum, since this organelle is involved, the rough endoplasmic reticulum is always engaged in the formation of proteins. And if I talk about this, since this organelle is not involved, this type of endoplasmic reticulum is involved in the synthesis of oil in our body, oil, lipids. What is the other word for oil? Earlier also for cell membrane I used that word, lipids. What was the composition of cell membrane I told you?
proteins
And proteins and lipids. And what was the one word we were using? Composition of cell membrane. Cell membrane is made up of that one word that I used. What was it? You will come in your book also. Cell membrane is made up of one word I use which I break up to say lipids and proteins. What is that one word?
What is that one word? No, composition only. What she says is correct. That lipids and proteins, but lipids and proteins when you club together to form a single. No, that is the structural aspect I told you, that it is fluid-like, not rigid. Lipids and proteins when you club together form one word. What is that word? Lipoprotein. Lipoprotein. Cell membrane is made up of lipoproteins. What do you understand by this? It has got 2 layers of lipids and 1 layer of protein. So that is your answer. It has got 2 layers of lipids and 1 layer of proteins. No? So when we talk about the endoplasmic reticulum, let us now learn what are some features, structure, function of the endoplasmic reticulum. This endoplasmic reticulum is in the form of a network of tubes. Endoplasmic reticulum. What is the meaning of reticulum? Reticulum means a network. Reticulum means a network. Structure, they are made up of a network of tubular structures starting from the nuclear envelope and sometimes terminating in the cell membrane and sometimes may not terminate in the cell membrane. And this is a network of tubes. Already I told you, it is a network of tubules. It is a network of tubules. And what are the 2 types? The 2 types would be, in short, I would write, it is RER. What can be the full form of RER? Rough endoplasmic reticulum. So what should rough endoplasmic reticulum contain? Endoplasmic reticulum plus another cell organelle. What is that other cell organelle I'm talking about? Ribosomes. Let us talk about the second type. The second type is SER. SER would be what? Smooth endoplasmic reticulum. So what is one difference between the rough endoplasmic reticulum and the smooth endoplasmic reticulum? The smooth endoplasmic reticulum does not have ribosomes at all. Functions, okay, cell types, where they are present. Sometimes you are asked, mention 1, 2 cells where they are present. If I talk about the rough endoplasmic reticulum, they are extensively present in the variety of different types of muscle cells. Do you know what are the different types of muscles in our body? Did you ever come across? No. If I talk about the muscles in our hands, remember, the muscles in our stomach, the muscles in our heart are absolutely different. Though they are all made up of muscles, but the muscles in our eyes and the muscles in our toes and the muscles in our heart again are not the same. So when I talk about all these group of cells have got a rich concentration of RER, has got a rich concentration of RER. So yes, I would say muscle cells, RER. And what about SER? SER, I would say one very good example of the SER is the liver cells. Liver cells have got a large number of anything you want to ask? Liver cells have got a large number of SER. Let us very fast go to the functions. The functions of SER and RER. Now, when I talk about the endoplasmic reticulum from the diagram itself, you get to see that it is a network of tubules spanning almost the entire width of the cell. So, one major function of this is it gives a structural framework. This endoplasmic reticulum gives a structural framework. If we are able to take out the endoplasmic reticulum from the cell, absolutely take out the entire network from the cell will squeeze. The cell will not have a definite shape at all. What next? When we talk about the endoplasmic reticulum, the first is this, I told you. Now let us talk about the specific functions of RER. What is the job of RER? RER is meant for synthesis of variety of different types of proteins. Now the RER is responsible for synthesis of different types of proteins only because of this particular organ. RER has ribosomes. Hence, it is capable of producing large number of proteins. And if I talk about the SER, SER cannot produce proteins because it does not have the ribosomes. And it can bring about the formation of lipids. Again, I ask you, what are lipids? What are lipids? Oil or the fat. Oil or the fat. So what did I tell you? SER is basically present where? Liver cells. So and in the liver cells, one particular function always happens is formation of the lipid molecule, formation of the fat molecules.
And these fat molecules can very well take place to carry out certain functions. We are yet to complete with the functions of endoplasmic reticulum. There are many more. You can note it down in your volume. We are still not done with the functions of endoplasmic reticulum. There are many other functions of endoplasmic reticulum that we'll have to discuss. Okay, we'll have to continue. We'll have to stop here because I think it's already time for your next lecture. I will give you 5 minutes break. I think 5:30 it is already now. We'll get 5 minutes break, not to worry. And after that, the next lecture. Okay, so 5 minute break.